No, I don't know. No, I'm saying it's. It's not a minnow.
Green hat, my dark hat.
I feel like you could. I guess you would have an opportunity.
Because I haven't been out in the field, so.
Yeah. I did that one. I was thinking of my other place.
Okay. Welcome, everybody. Welcome to those of you online that have joined us for week 7 of our Stone Lab Thursday night seminar series. We are very pleased to host one of OSU's own, a relatively new addition to the School of Environment and Natural Resources. Dr. Allison Rober is going to join us. She comes most recently from Ball State University, but is doing some very exciting stuff on microbial ecology and cyanotoxin work in Alaska wetlands and elsewhere. And I'm sure she's going to wow you with all things microbes today. So without further ado, please welcome Dr. Allison Rober. She'll give her presentation and have plenty of time for questions later. So Allison, take it away.
All right, well, thanks for having me. So I'm Allison and I'm an aquatic microbial ecologist. And today I'm going to share some of my lab's research examining environmental controls on cyanotoxins, which I'm going to pretend like none of you know anything about. So, so cyanobacteria are a type of algae. So I am traditionally an algal ecologist. And so some people call cyanobacteria bacteria because phylogenetically they are, and I'll get into that in a little bit, but they also get studied by all the algae people. So just like the rest of the algae, cyanobacteria grow by photosynthesizing and converting water and carbon dioxide from light energy into sugar that they themselves use for growth and development, and then release oxygen as a byproduct. So we can all say thank you to the algae for about every third breath that we take. Should I just admit people as they come? Okay. But in addition to the compounds that are used for growth and development, most species of algae release a fraction of their photosynthetic products directly into the water column. So we might just call this dissolved organic matter, right? It's a big term that can be lots of different kinds of carbon and things like that. So in some species of cyanobacteria, not all cyanobacteria are bad, but in some species, the exudate pool can include a suite of toxic compounds that are harmful to human health. So microcystin, for example, is the one I'm going to talk about today, and is most familiar to probably many of you here, is among the most commonly occurring toxin produced by cyanobacteria. And it's a hepatotoxin, so it causes liver complications and damage to the nervous system if it's ingested. And so these are the limits set by the World Health Organization. 1 microgram per liter in drinking water is allowable, 20 micrograms per liter for recreational use. In the absence of nutrient limitation, temperature is often considered the most important determinant for the growth and metabolism of freshwater algae in general, including cyanobacteria. And that's largely because cyanobacteria has a higher temperature optimum growth rate. So we can see here in this figure compared to the other larger groups of algae. So we put them in big functional groups. Green algae are the chlorophytes, dinoflagellates or diatoms. They have lower temperature optimum growth rates. And so cyanobacteria tend to be competitive at higher temperatures. And so the typical photosynthetic response of cyanobacteria to temperature is this progressive increase until they reach their physiological optimum. But because toxins are produced with photosynthesis as a product of photosynthesis, higher temperatures also result in higher toxin concentrations. And so because of that, warming generates this positive feedback on harmful algal bloom development. So first, it promotes toxic cyanobacteria strains over those that are non-toxic. And then it stimulates toxin synthesis. So at warmer temperatures, more toxin gets produced because the metabolic rates are higher. And then in some of the research that we've done in my lab, we have found that at higher temperatures, the toxins are subsequently released from the cells. So they're purged into the water column. And this then sometimes, some research suggests that the toxins are used as a signal to the other cyanobacteria to upregulate toxin synthesis. So it can cause this, well, it's called a positive feedback loop, but it is quite a negative thing to kind of keep producing toxins and keep getting more toxic. So this is particularly problematic because, oh, sorry, I missed my little circle there. So this is really problematic because surface water temperatures are continuing to rise globally, right? And so in fact, blooms of toxin-producing cyanobacteria have increased in frequency and severity with environmental degradation of freshwater ecosystems across the world. And so this is a really figure from 2024 showing trends in lake surface water temperatures. So we can see quite a bit of warming trend. And then more closer to home, this is was just published this year on the Great Lakes, and anomalies is what this is showing. So unusual warming trends. And so we can see that this is, it's already a problem here and it's likely becoming worse. But one interesting thing is that cyanotoxins don't typically accumulate in the water column long term. And so, but we know from lots of research that toxins are really difficult to break down. So UV light does not degrade them, chemicals don't degrade them. Temperature, they're really resistant to denaturing. And so one hypothesis is that microbial degradation plays a role in this removal process. And so biodegradation is just the microbial breakdown or biological breakdown of cyanotoxins by heterotrophic microbes that live in association with the algal bloom. So the sort of scientific jargon is to call it a phycosphere, or the microbiome that surrounds the algal bloom. And biodegradation of cyanotoxins has been widely reported in freshwater ecosystems. So this has been shown to occur, but there's really been little investigation into the factors that regulate microbial mediated degradation. And so just like cyanobacteria, heterotrophic metabolism is sensitive to changes in temperature. So elevated metabolic rates occur at warmer temperatures. So here I'm showing some data from another experiment with warming, where it's just showing bacteria density over time in ambient and warmed conditions. And you can see that there's a lot more growth in the warmed treatments. And in order to maintain metabolism, bacteria need to consume organic matter. And we see that if we feed bacteria dissolved organic matter, they will consume that carbon at a much faster rate with warming because their metabolism is higher. This is why you put your food in the refrigerator, especially fruit, right? You're trying to slow microbial metabolism, consuming the sugars in your food. So the idea here is linking this back to toxin producing cyanobacteria, that owing to these increased metabolic rates with warming and higher temperatures, that microbial mediated degradation may be influenced by warming. And so now getting back to the toxin I'm specifically interested in, microcystin, which is this whole molecule here, is among the carbon compounds that's released by algae, as I mentioned, during photosynthesis. And many bacteria are capable of degrading microcystin via a specific gene. So MLR is a metabolic pathway where they can break the bonds of this molecule into, that's what these different colors represent. So they get broken down and turned into a linear molecule as opposed to a ring that's really hard to degrade. And then they can be consumed in their smaller constituents. So because heterotrophic bacteria have higher metabolism at warmer temperatures, and this molecule has carbon attached to it, the idea was that microbial mediated degradation may be influencing or influenced by warming. So with that in mind, the goal of the study that I'm going to talk about today was to evaluate the ability for heterotrophic bacteria to degrade microcystin across a natural temperature gradient. And so the hypothesis that we tested was extracellular toxins, so those that are actually purged, not in the cell, release or stimulate heterotrophic metabolism and then the subsequent degradation of the compounds. That cyanotoxin degradation is enhanced by warming, and that cyanotoxin degradation is regulated by bacterial community composition. So to begin producing, we produced toxin-producing cyanobacteria were collected and condensed in a plankton net. So this is one of my former students who's, all of this is a monoculture of Planktothyx agardii from Grand Lake St. Mary, which is here in Ohio and has year-round blooms of Planktothrix even under the ice. So we collect tons and tons of water and condense the algae. And then we convert all of the liquid to a powder using liquid nitrogen and attaching it to a freeze-dry unit. So these are some of my students. They're playing with liquid nitrogen. So we freeze all the liquid and roll it around and then attach it to this freeze-dry unit. And this is what you get at the end, is the powder that's left after all the water has been dehydrated. And this allows us to then use this exudate powder for experiments. It can be rehydrated for experiments where you can manipulate the concentration. Now, because we took this from an actual natural community, the extract contains the toxin, microcystin, but also a lot of the other dissolved organic matter that would be part of a natural microbial community, proteins and nutrients and things like that. So we manipulated, this was a laboratory incubation experiment. And so we manipulated seasonal variation in lake water. And it was manipulated at, we used the temperature range 5, 10, 15, and 20 degrees Celsius. And we did this in a recirculating water bath. So we could control the temperature. And they were plumbed to these jacketed beakers, which is what this diagram is showing. So water circulates around and there's water inside of here. And so it passively alters the temperature of the bottles inside. And so we had these all connected in sequence so that we could have a different water bath for each water temperature treatment. And we chose these temperatures because in our previous year-round sampling so we sampled cyanobacteria blooms under the ice year-round, and that represented the natural range in water temperature that the cyanobacteria experience. And so we were really interested to find out if heterotrophic bacteria could degrade toxins along that same temperature gradient. So we used that again. So water for our experiment was collected from Grand Lake St. Mary, and then we filtered that through, most of it through a 0.2 micron filter into sterile flasks. So 0.2 should remove most everything, even bacteria that typically don't pass through a 0.2 micron. So then we added our rehydrated microcystin extract, and then we filtered a portion through a 0.7 filter, which would capture all the cyanobacteria. So we weren't adding any additional, but it would allow a lot of bacteria to go through. And we use that as our bacterial inoculum at the start of the experiment. And so then this whole experiment was conducted in the dark because we didn't want any photosynthesis to happen or any cyanobacteria there. We wanted just to look at the heterotrophic bacteria's ability to degrade the toxins. And so then we measured microcystin concentration, bacterial abundance at time intervals. So we did an initial measure at time 0, after 6 hours, 12 hours, 24, 48, 96, and 192. So a total 8-day experiment. Our microcystin concentrations were measured using a high sensitivity enzyme-linked immunosorbent assay, ELISA kit for short. Our bacterial abundances were determined by direct counts. So we stain the bacteria cells with DAPI, which makes them glow, and then you can count the bacterial cells. And then our bacterial community composition was determined from samples that we collected at time 0, which I'll refer to as initial, and then 8-day community. So we just sampled the bacterial community beginning and end. And the bacterial community was determined with DNA extraction and metagenomic analysis of the V4 region of the 16S rRNA gene. So that's kind of the standard molecular approach for determining bacterial community composition. So these are some of my students participating in all the data collection. And what we found is that microcystin concentration, so I'm going to click these in. My key here are the different temperature treatments over time. So first we're looking at microcystin concentration. And so we see that microcystin declined over time at 5 degrees. It declined a little faster at 10, declined a little bit faster, but not significantly different at 15. And it declined even faster at 20 degrees. And our patterns of microcystin degradation corresponded with increases in bacterial cell density. So we see a slight increase, especially towards the end in our coldest treatment, bacterial increase at 10, 15, and 20. What then, from our molecular analysis, we see that bacterial community at all temperature treatments were different from the initial bacterial community as well as from each other. So the way I'm interpreting that is, so this is a principal component analysis. And what we're showing here is these axes we won't get into right now, but as things are spread out across from each other, the farther they are apart, this is a measure of dissimilarity. So if things were all stacked right on top of each other, that would mean the community was the same. And so we can see here, this is my initial bacterial community. So this is time 0. And you can see cyanobacteria. I've put some of the major phyla that were prevalent in my samples here. So the community initially was almost all Planktothrix. And then we can see that these are our temperature treatments, and they cluster very far from the initial community, but also from each other, showing that there's actually a distinct bacterial community at each temperature. And we can also show this another way. So this fancy heat map. So let me explain this a little bit. So heat maps, we're reading the colors. The hot colors mean a higher abundance, and the cold colors mean a lower abundance. And we can see the same clustering happening here. So here is my initial lake water sample. And these colors over here are the phyla that are over here. And these are all the genera that you might see. And I know you can't read those real close, and that's okay for now. What we're really interested in is this clustering. So we see a really high hot colors up here for these, and they happen to be the cyanobacteria genera. And then at the next temperature, we see that these are the genera that are really abundant, and those are not prevalent in the other treatments. And we see the same thing at 15, and then we see a whole bunch of bacteria are really abundant at 20 degrees. But there's no overlap among these genera, which is really neat. So there's this, starting from the same exact initial bacterial inoculum at each temperature treatment, is selecting for a different type of bacteria. And now we did not probe for specific genes, though I have the samples, so we could do that. But many of the species that we found here, genera, are capable, have been shown to possess that MLR gene. I didn't look and quantify if the gene's present there, but many of the genera have been found in the literature to contain that gene. So, and then we think that declines in relative abundance. So this heat map is also a measure of how relative abundance to each other. And when we see a decline in some species compared to others, this suggests this trade-off, a physiological trade-off with temperature tolerance and competitive ability with other bacterial strains. So kind of putting this all together, so patterns of microcystin microcystin concentration declined over time, and that degradation increased with warming. And those patterns in degradation corresponded with increases in bacterial cell density, and they were also enhanced by warming. And then we saw this distinct bacterial community in each treatment that corresponded with differences in our microcystin degradation. So taken together, these findings suggest that this observed decline in microcystin that we saw was influenced by microbial mediated toxin degradation that increased with rising temperature. And these then support our hypothesis that temperature and bacterial community composition mediate cyanotoxin degradation. And now, sort of relating this to some bigger ecological questions. So a key question in ecological research is whether biodiversity is important for ecosystem functioning, right? So it's widely assumed that the role of different bacteria in an ecosystem function is related to community structure. So, and the more diverse the community is, the more resilient that community will be to change. And contrary to our expectations, these are actually species richness, so the number of different kinds of bacteria we observed in our different treatments. We didn't actually see the highest degradation in the treatments that were the most diverse. Instead, we think that it was really degradation seemed to be more of a function of bacterial abundance of a few dominant species within each treatment. So to give you an example, in the purple here, this was the greatest rate of microcystin degradation. And in that treatment, there was nearly half the number of species that was observed in the initial community. But despite these reductions in species richness among treatments, this community, what's really fascinating is that it maintained a population of bacteria capable of degrading microcystin across the entire temperature gradient. So this suggests a degree of functional similarity in the ability of bacteria to degrade microcystin with changes in water temperature. And the presence of these species with overlapping functions should buffer the system against individual species loss and promote some resilience to an ecosystem experiencing a harmful algal bloom. And so the degree of functional similarity that we observed here suggests that microbial degradation will persist in the face of rising global temperatures, which is kind of a good thing. Silver lining. And so also what was interesting is irrespective of taxonomy, the degradation ultimately reached a very similar end concentration. So across the treatments, microcystin was reduced to just about the 1 microgram per liter threshold, right? So this is what's safe for drinking water. And so we saw this significant reduction in microcystin in all the treatments, irrespective of temperature or community, over 8 days. And so this information is useful because for ecological purposes, but also water treatment. And so biodegradation is a low-cost way, right? Bacteria just do this for free to reduce microcystin concentration prior to potentially more expensive water treatment options. Now that we know there are some bacteria that are more tolerant at different temperatures, there could be possible inoculation with bacteria capable of degrading microcystin at different temperatures. But ecosystem consequences, so we know there's increasing frequency and severity of toxic blooms with rising temperature, and that water temperatures are expected to continue to rise. And so microbial degradation should persist under these conditions, is what the data from this study show, and that it's likely going to play a pretty significant role in trying to maintain some kind of ecosystem health under rising water temperatures. So that was actually really fast. So I want to thank all my students and my collaborators and funding sources. And I'll take your questions. Yeah, Kate.
I was wondering, I know you're talking about like the implications of this study. I was wondering if you looked more into like how those like microcystin fighting bacteria could be like implemented in a water quality standpoint and like to combat these the harmful algal blooms.
Yeah, so I think are you kind of thinking like a water treatment facility?
Yeah, and like I know you mentioned that in like the last 2 slides.
Yeah. So the thought there is, so too, a lot of like retention ponds at water treatment facilities can be like, it's, subject to environmental temperatures and get really warm. So that was sort of the idea is if you know that there are certain bacteria that can tolerate other temperatures, if they're not naturally there, that might be something to consider is like looking at the bacterial community and selecting for those that could still perform that function at a higher temperature, just to kind of get it down to a reduced amount before other folks here are researching other ways of getting rid of toxins. So, yeah. Yeah.
In the extract that you made, the powder material, I might have missed something. That's kind of just like the whole cells that have been dehydrated, or is it cells that have been lysed first? I mean, I'm sure if there's filtered material, or just the whole cell. It's
Lysed likely in the freeze drying process, but yeah, it is the intention was to create an extract that did have all the natural constituents and nutrients and things like that.
So I was wondering is if you had any other measurements in the experiment, say looking at just the the use of all of the organic carbon or maybe measures of bacterial growth to reproduction or metabolism to sort of show, did the microcystin get used more preferentially than the rest of the material at different temperatures?
Yeah, so so I didn't break apart a lot of, but this is dissolved organic carbon, but I didn't break apart the different kinds of sugar. And it actually starts to increase, which is something we see a lot in these experiments as there is cell death. It actually contributes to the organic carbon pool, but they weren't necessarily taking it up like you would expect if they were like giving them glucose. And this SUVA is a measure of how recalcitrant or hard to degrade the carbon is. So the higher it gets, the more difficult the carbon is to break down. So carbon's increasing and it's getting more recalcitrant towards the end. And these are nitrogen and phosphate concentrations. And so we can see that nitrogen really started to get taken up kind of towards the end of the experiment. But I guess in my experience, I wouldn't call this severely limiting yet, like that it was, and phosphorus got pretty low by the end there. So it seems like nutrient limitation was not driving our trends, I guess I would say, especially because they all got to the same microcystin concentration, just at different rates. So, does that answer your question?
Yeah, I was just thinking like it would be a very fascinating result if at certain temperatures microcystin was preferred.
Yeah.
Maybe related to the changes in the microbial community that you observed at those.
Yeah.
But yeah, it's
No,
hard
that's neat.
to kind of parse that out.
Yeah. Yeah. Thanks. Yeah.
So just for clarification, like with global rising temperatures, is that in reference to like seasonal changes or climate change?
Yeah, well, so it's related to climate change. And so for example, every summer has been the hottest summer on record, sort of globally, but our globally water temperatures are rising. So the warmers are getting warm. And in the previous work that I've done, the reason we found 20 degrees, at least with Planktothrix, to be a threshold. So but anything above that, which is like 70 degrees Fahrenheit. So that's pretty commonly met. Planktothrix will purge their toxins. They become really stressed at that temperature. And so they just start dumping their toxins into the water. So it increases toxicity. So that's kind of the feedback, yeah.
Go ahead.
When you collected the lake water for inoculum, what time of year was it? Then part two, my question is, but if you were to grab Grand Lake St. Mary's water at 5, 10, 15, 20, would you expect what would be naturally occurring in the lake match what you would find in your experiment after it things?
So to the first one, I believe it was late March. It's been a while, but it was kind of early spring. I wouldn't expect the community to be exactly the same because this experiment was completely darkened. So, and if it was those temperatures in the natural lake, then the cyanobacteria I guess if we were just looking at they would still be in the 16S analysis. So you'd have to remove the chloroplast to really get a sense if the community would be the same. But I suspect that you should, because that was the initial bacteria, the initial sample was the inoculum for everything. So.
There was a question in the chat. Would you get similar results in a different body of water in a different region?
I think so. So I'm curious to know. So that's kind of one of my questions from this to kind of keep building on this is this Planktothrix specific? So a lot of the literature, there's a bacteria called Stingomonas that is really common with microcystis blooms that wasn't even present here. So it's one of the first bacteria that the MLR gene was found in. And we did not even find that in this at all. So it's kind of interesting to me that the ones that get published the most, we found some new, this bacteria, no, it's a colobacteria, this purple bar here, hasn't previously been shown to be a microcystin degrader. It's often in association with toxic blooms. And since we didn't look for the gene specifically, I can't say for sure that that's what it was doing. But it was the one that was, the relative abundance was a lot higher at that temperature. So this may be new information as well that is specific to either this type of algae, like this phycosphere, or that lake. So we could do it here and find out. Yeah.
Is there a way to regulate or possibly increase those bacteria levels in like a 5-hour liquid here?
I don't know. Yeah, I mean, I'm not sure you would want to. I mean, yeah, I don't know. That's tricky. That sort of gets into like biological control, which always makes me a little bit concerned, but yeah.
So you mentioned like when the cyanotoxin like under heat stress, they like purge this microsystem. What's the actual function of the microsystem? Is it like a waste product, or is it like a lubricant or something?
So that's but that's not actually known. There's been a lot of folks have proposed that it's to prevent herbivory, but research doesn't really support that. But newer research is showing that it is a stress response. Because this is the same thing actually happens in places where sea levels rising and salinity increases. It causes the same cellular stress, and those those toxin producing algae will purge their toxins. So the sort of the newer hypothesis, and there's more evidence to support it, is that the molecule is actually to tell other cyanobacteria that the conditions are getting stressed and they should upregulate their toxin production. So kind of like trees will put out a bunch of seeds when they're about, really stressed and think they're dying, they'll start to reproduce. It's supposed to be a stress signal. But that I haven't actually explored myself. So I haven't looked for like stress response proteins or anything like that that might actually provide evidence for that.
Okay, let's give Allison another round of applause. Might go too far,
Okay.
because I've got to pull up something else, but
Okay.
while you're here with us, let me make sure okay, thank you for the chats, questions. Would you mind telling everyone here how you got into this field? What got you excited about doing science?
So I did my undergrad at Michigan State, and I went there because I thought I wanted to go to vet school and study horses, like probably most little girls. And then in my first semester, I realized that is not what I was passionate about. And I realized what I was passionate about, because I'm from Michigan and the Great Lakes state, that I was very passionate about water quality. And so I started working in a lab who ended up being my PhD advisor, Dr. Jan Stevenson, who is an algal ecologist. And he used algae, specifically diatoms, as indicators of water quality. And so I got a job in his lab, learning to identify diatoms on the microscope and using them to infer water quality in the Muskegon area on the west side of Michigan and Lake Michigan. And he also did a lot of policy work, taught an environmental policy class that I was fascinated by, because he did a lot of work with the EPA, and they used his work as expert testimony in court cases on water quality and trying to get people to be held accountable for non-point source pollution. And so I just was fascinated by all of that, and I decided I was gonna pursue graduate school in his lab. And so I started my graduate program. I started as a master's student, and then I actually met my husband in the same lab. Obviously he wasn't my husband then, and so we were both into algae, which what are the chances? And so then we started a research program of our own in Alaska, where we do a lot of work still today. And so I continued on as a PhD student. I just turned my master's into a PhD, and here I am.
Thank you for that, Allison. Those of you online, and those of you in the room, if you would like to share with Stone Lab and Ohio Sea Grant, if you've gotten anything good out of this seminar, and it's going to change your life completely and turn all you hoodlums into nice, outstanding young people. I kid. Please take our questionnaire and show Congress and everybody how much Ohio Sea Grant seminars matter to you. Just a little plug there. So thank you again, Dr. Rogan. Justin,
Right, so next week, we're starting at 6:30.
yes, 6:30 next week. So,
The RU students starting to go.
yeah, those of you on Zoom, we will be,
Presentation, 4:20 minute presentation.
yep. Those of you on Zoom, we're starting at 6:30 next week, and it'll be our 4 RAU students presenting their work research from here at Stone Lab this summer. Thank you, everybody, for coming and joining on Zoom, and have a good rest of your evening.